morning, everyone. Welcome to another insightful sessions of the Breakfast UMHealth Live webinar segment. I'm Soon Hao, Senior Lecturer at the Department of Biomedical Science, and I'll be your moderator for today's morning sessions. We are truly privileged to have with us Dr. Kim Kee, who will be speaking on a very timely and critical topic, pathogens detections to outbreak response. As we all know, effective pathogen detections and response strategies are pivotal in managing infectious disease outbreak. With global health challenge becoming increasingly complex, this topic could not could not be more relevant to us. So allow me to briefly introduce our speaker. Dr. Kim Kee is a senior lecturer at Department of Biomedical Science. Her work has focused extensively on infectious disease diagnosis, pathogens biologies, and outbreak surveillance strategies. And now, without further ado, I would like to invite Dr. Kim Kee to begin her presentations. Dr. Tan, the screen is yours.
Okay, good morning, everyone. And thank you, Dr. Soon Hao, for your kind introduction. And I'm Kim Kee. I'm from Department of Biomedical Science. Today, I'm going to take you on a very short journey from pathogen detection all the way through outbreak response, with a special emphasis on genomic study or genomic surveillance. Okay, let's start with a chronological look at emerging outbreak over the last 2 decades. From the green dot, you can see this is an indication of a new pathogen when they were first described or reported. And those red label is used to show an existing disease where they have appeared in a new country or region. So this timeline briefly tells us 2 things. First, the novel spillover or an infection involve new pathogen occur regularly. And the second, that it's even a known pathogen is currently continue to expand into a new territory. And when we have the same data that we put on a world map, we can see that the emerging outbreak occur everywhere, not just in one hotspot. Also, when you look at the map, you notice that different area or region will have a different diseases. So the surveillance and the response for every country cannot be one size fit all. So for this, we not only need a global coordinated network to actually detect the threat at their source and maybe from different countries, but a local talent team that we have our own diagnostic and response strategy should be in place for us to better react when we have an infectious threat that hits us. And data shows that roughly 60 to 80% of the emerging infection are actually derived from animal source. This is pretty general because human and animal have actually coexist for a long time. And in recent decades, our interaction with animal have changed and increased. This has increased the chance of the animal viruses, especially those from wildlife, jump over into people. However, when we spot a virus in an animal, an arthropod, or even those viruses that are present in humans, does not necessarily indicate that that will be a human epidemic or there will be an outbreak. For every successful emergence of this new virus into the new population, such as human population, it needs to have essential steps. This involves the first one, we call it a spillover. Is when an agent is actually introduced into this new population and new host and start its first infections. And second, the agents need to be able to establish, means that it able to establish and then it transmit within the host and the population itself. So in this slide, we focus on spillover events. It's when this microbes first jump from the animal reservoir into the human and it have its first infection. So on the left here, this is a very simple spillover events that scientists used to think, where we have a very simple framework. Is like you only have one species and that species carry a particular pathogens, and the pathogen will infect our people. And then even though, this host that usually you can't really identify them, but once we able to ID what are the hosts, and then we will be able to trace it and then to study the spillover event easily. But in reality, this is not the case. In reality, the spillover events is far more complex than what we think. You look at these diagrams. So normally there are multiple hosts will be involved in the wild and in the environment, even before the pathogen manage to reach us. And then there could be multiple pathogen cycle within the different animal species, the mammal, the birds, and sometimes involve the arthropod could happen. And for this new pathogen, so emerge into the human population, at the very beginning, the animal, the arthropod species that truly, involved in this transmission is rarely clear. So making the surveillance, the study about the risk become very challenging. And even now, I think COVID, everyone know about it. We still don't know what is actually the host and then the reservoir for this disease, even though there has been with us for a few years already. Furthermore, excuse me, multiple hosts can carry different virus, and a host can carry multiple virus. So that actually makes the identification of these spillover events become more and more challenging. So after, the pathogens has its first infection in the humans, in order to sustain the infection in the human population, the pathogen has to be able to gain what we call transmission fitness, that it must be able to overcome the within host and then the population level barrier in order for it to spread. So post the spillover transmission, when the virus go into the human population, it can have several outcomes. So the virus go into the human population and then it go nowhere. So there's no spread. So this is because that maybe the virus cannot replicate efficiently in the humans, or our immune system is actually killing the virus. So it cannot have any sustainable infection after this. So this is just end like this. So the other scenario, it will be, you will see that the virus can replicate in humans, and then it can have certain infection and it can have certain transmission, but it cannot have a large transmission or very efficient transmission in humans. So the outbreak is usually self-limiting. But there will be another scenario is when the virus adapt very well, such as like influenza, like SARS-CoV-2, like Ebola. So it actually crossed a critical threshold that, one-to-one transmission. So now a single case can infect many people. So that's when the epidemic and pandemic that is really can happen following, the spillover and transmission by this virus, which adapt into human population very well. So when you look at here, the stage of the emergence is when you have a virus and it start to adapt into the new host. Here we talk about is human population. It can be any animal population as well. And then when it become very effectively in replicating and transmitting, then it will be able to increase the number of disease to a certain extent to an outbreak or epidemic or even a pandemic level. But when that particular pathogen, it become constantly present, it no longer increase the number dramatically. So it coexists with that population already. So it enter into an endemic stage. So you always will see the virus or the pathogen and present together with the people. So there's always a small outbreak and comes in that consider endemic stage. And this one, when that particular microbes, it become no longer pathogenic and it become beneficial and it could be under this fully adapted stage. It's not necessary that every microbes it will go into this fully adapted stage eventually. So this is just a hypothetical scenario that in the stage of emergence that pathogen could occur. So So when we have this detection of this spillover, it's not automatically saying that now you have this animal pathogen that present in human, and it is an indication that there definitely will be an outbreak. But a detection of this viruses will give us something that you need to do something. So there is a detection for an action. So a lab confirmed in that case is actually telling us that, hey, yes, we should start to look for is there any possibly ongoing transmission, especially those pathogen with a pandemic potential means they can adapt in human population very well, and they can easily be transmitted, and it can cause a large outbreak. So those pathogen with this characteristic, when we have the emergence of this pathogen, and then we should pay attention to it and confirm if there is any active transmission point, and then also looking at is there is any population at risk, that after that, and then we need to check on the groundwork, including check on the ecology, the reservoir, the arthropod, and to consolidate all the evidence that with that, and then we can do a full outbreak response. So in short, the detection of this spillover event is a spark. It's not just a fire. It's not the actual fire, but it actually kickstart that we need to do something to do investigation in order to have a proper control. However, for a new pathogens or an uncommon pathogens, our first line defense or routine that in the healthcare system is normally the diagnostic. But for this new pathogen, they are not effective because our healthcare system diagnostic is the standard assay that is not designed for detect something entirely unfamiliar. So even like when we do a broader surveillance in the central laboratory, and you're using serological assay, it's more robust, it's more general, or a PCR panel that you can detect for many of the pathogen. They can tell us when it can pick up this infection, they can tell you that this infection is happening. But normally those information will not be sufficient to tell you, where exactly and then this pathogen come from, and then whether it changed. And then the true identity of this pathogen down to the species or genotype level, even the clade level, they will not be able to review just by the normal standard diagnostic or detection assay that we use. So therefore, we need an integrated multi-species surveillance plus ecological. And this framework, we call it a One Health approach because we see majority of the outbreak or emerging disease right now is from the animals. So a One Health approach that actually consider both human, animal as environmental into a single approach, that will be important. And then with that, we incorporated with a genomic surveillance where we can fill in the gap by reading the entire genomic genome of the virus itself. So it allowed us to actually pinpoint the origins and tracking how the virus could have spread from the animal population into the human population, and show where are the adaptation, and maybe you can find, where are the meaningful mutations. So for today's presentation, we will use flu influenza virus as an example. This is because influenza virus A is carry both a burden for a seasonal flu as well as it is a potential threat for the next outbreak. We will briefly explain this later on. And then so it's a very good example that we can use to show you why it is important to use One Health approach as well as genomic surveillance to do a investigation or a preparedness for a potential pandemic in the future. So influenza virus, so there are 4 types of influenza virus. So the virus is an enveloped segmented negative strain RNA virus. So out of these 4 influenza viruses types, and then only influenza A and influenza B are currently cause seasonal epidemic in people, what we call them a seasonal flu. Okay, C and C can cause infection in human, but it's usually mild, so it's not really involved in any of the epidemic. And D is normally cause infection in animals. So to date, it's out of the influenza A and B is only influenza A is known to cause a flu pandemic. And this is a very busy slide, but actually it only tells us one thing. A major influenza pandemic is normally arose while antigenic shift means there is a resortment of the genome segment of different strain, as you can see here. So this resortment of a different genetic strains could happen, and it will create a new or novel combination that allow this new strain that effectively invade into a new population. In this case, is human. And this reassortment can only occur when these two influenza virus were actually infect into the same host. So during the co-infection into the same host, and then during the virus replication, reassortment of their genome segment could happen. When we look at the influenza virus transmission cycle, that you can maybe understand this one better, that the influenza virus is actually naturally circulate in the aquatic warbird. So this is their reservoir. And then they can spill over into other mammals such as poultry, pigs. And in this intermediate host and co-infection of different influenza A virus could happen. And that is where all this mutation and reassortment could happen and create a new novel strain. This strain, when they pick up a suitable adaptive mutation, and then that could actually boost their replication and transmission in the mammals. And of course, that includes humans. But while in humans, when we talk about flu infection, we usually have to think about two different transmission patterns. The first one, when we is about subtype like H1N1, H3N2. When you talk about this, they are the subtype that already have an established ongoing transmission chain in people that can cause unpredictable, the yearly epidemic, the seasonal flu, that as we can see here. But there is another one when we talk about flu that will be sporadic avian origin spillover. This normally involve H5, H6, H7, H9, H10 viruses that typically will actually infect animal population, but they gained the potential and then to spill over into human. But normally it doesn't really involve many people for this infection. But what worrying is what if they infect human and then through that they get the adaptive mutation that allow a human to human transmission. And that actually carry a very significant pandemic potential. For now, there is an ongoing spread of H1N1 strains in the poultry and the wild birds. And then this is what is worrying because it has the increased mammalian transmissibility. And for this H5 infection, as mentioned just now, and then it is not something is new, so it's have been detected in late of 1950s. Over 20 years, it has caused an infection in more than 800 of human infections in 23 countries. But over the past years, since 2024, we have seen this H1N1 clade 2.3.4.4.2, 4.4b in the US dairy cattle, especially also it infect poultry as well as the farm worker. And this is a very worrying sign because this is an indication of that this lineage could have started adapting into the mammals. And that laboratory study also shows that it carries mutation can enhance the transmission fitness. You know, when the combination of it will spread into the different animal circulation in different animal species, as well as when it have a mutation that can enhance its replication in the mammals. So that's where it become truly worrying that it could become the next candidate for a pandemic. So here is where exactly a One Health approach coupled with genomic sequencing should come in, and it is actually ongoing. So by rapidly decoding the genome sequences from various sources, they collect sample from the animal, collect sample from the environment, collect sample from the infected humans. We can start to track, you know, where are the source and how the virus being transmitted, and then when they emerge, and then how they move. And with that, they can also become a valuable information to feed into our diagnostic, antivirus choices, and the development of vaccine that to prepare for this pandemic. So with that, we're closing a gap between we found it and then we can stop it. And by using this One Health approach as a surveillance method, as well as using the genomics to provide us the information to design an effective diagnostic as well as therapeutic. This is just an example that what have been done. So this H5N1 clade 2.3.4.4b is the predominant clade that's circulating in 2024 and 25 by using this One Health approach and coupled with genomic sequencing. So we can find that it is not only one, but at least two particular genotype are actually co-circulating. So those are the B3.13 and D1.1. Also, with the integrated information, we also manage to link back the human cases to their animal hosts. And with that, we also be able to find out the molecular marker that indicates a host adaptations, as well as we can use the genomic informations of the flu that to monitor whether those strains will be able to escape the current treatment or not. So this is very important because that if there is any antiviral escape variants and the intervention should be very quickly come in. So with those information all come together, and then we will be able to do a more proper pandemic preparedness to prepare if there is any potential outbreak in the future. So on my last slide, so it's just a take-home message. So detection of a spillover events is very important, and that could be acting as a starting for us for any of the downstream investigations. But human-centered surveillance is no longer sufficient. So an integrated surveillance system by using One Health approach by considering the animal host, the reservoir, and the environment. And sometimes we should take into consideration arthropod as well. And then it will be a more well-rounded system for us to be able to catch and stop the next pandemic. And of course, last but not least, a country level genomic surveillance is critical. It is not only part of the surveillance system, it is also important tools that we will be used for effective therapeutic and vaccine design. With that, I would like to say thank you very much. I welcome for any question. Thanks.
Okay, maybe I have this simple questions here. So, how do we ensure a quick response when an outbreak occurs, particularly with the limited resources like in Malaysia?
That's why actually I think COVID actually have demonstrated a very good example. Even we have a very limited resources, but as what we can see, we have actually did very well in our COVID pandemic response. What happened during COVID pandemic was like when we have a limiting resources, what government did is like we consolidate all the possible resources that in the country itself, including they get help from university. And in fact, in my laboratory is one of the laboratory who actually which did the sequencing then for the country COVID-19 pandemic surveillance, which helped the country to track the mutation and the variants present in Malaysia. That's where when we have limited resources, we cannot only rely on MOH to do that. So they were actually using all the resources that in the country itself to help the country to actually do the surveillance and help the country to get sufficient data. But at the same time, of course, we have already boosted up our genomic surveillance in the country level, where they start to train more of the parallel laboratory and empower them to be able to do sequencing. At least for now, and some of the hospital, for example, like Ipoh Hospital, they already have the capability to actually perform the sequencing at the hospital itself. So that's where when we starting from COVID-19, but I would say if there's a next pandemic comes in, and then definitely with a genomic surveillance that have been decentralized into many of the hospital right now, especially the hospital that do a lot of diagnostic, and then I think that will be definitely helping the country to do a very quick response and pick up, I mean, the variants more quickly. But still there's a gap, but I think we are currently improving.
Thank you so much. I think it's time. So as we need to wrap up today's sections. So I'd like to extend our heartfelt thanks to Kim Kee for sharing her time and expertise with us. Now I would like to pass over to Prof. Fong for the second episodes of Human Health. Thank you.